Next up, we have Johannes Sake, who is at Trinity College in Dublin. Now, his research focuses on membrane transporters in the pulmonary epithelium and their influence on the disposition of inhaled drugs, with the aim of getting a better understanding of absorption and clearance mechanisms in the lung. He does this to assess the interaction of transporter proteins with clinically approved drugs for inhalation, and today he's talking about the generation of a new organotypic cell knockout model. Take it away.
Thank you very much. Good afternoon, and thanks for inviting me to give a talk about my PhD work, which is about pulmonary MRP1, that is a transporter in the lung epithelial barrier, and he talks about the generation of a knockout model. What is our area of interest? We're looking at lungs, of course, human lungs in most cases, and here we have different absorption barriers. The bronchial epithelium is very different from the alveolar barrier, and we are interested in how agents, compounds get from A to B across this barrier. That can be anything that is inhaled, for example. How does it cross into the tissue and potentially further into the systemic circulation? There's different mechanisms involved in this, and if we zoom in a little bit further, look at a single cell, for example, we can see passive diffusion. It can be channel facilitated or transporter mediated. Now, what are transporters to begin with? Just proteins in the cell membrane facilitating a transport of compounds against a concentration gradient. And if it's energy dependent on ATP, then these transporters are put in the group of ATP binding cassettes. A very well-known transporter in this group would be PGP, but its relevance in the lungs isn't as high, so we are focusing on one called MRP1. What is the current understanding about drug transporter interactions? We have more than 450 transporters identified in humans and many compounds interacting with these. And in 2018, for example, 69% of newly approved, FDA-approved drugs showed transporter interactions, even though they only tested a handful of transporters here. And why is this important? Because a patient potentially taking multiple drugs interacting with the same transporter might encounter clinically relevant side effects, where concentrations of one or both drugs might be too low to reach the therapeutic window, or too high and would reach a toxic effect. And because of this, companies now have to test for these interactions before approval during the development stage. There's just one problem. This doesn't apply for inhaled drugs. They don't have to do this if it's inhaled. But this might change in the future, and because of this, we and companies are interested in investigating this. Back to MRP1, the transporter we are looking at. MRP1 stands for multi-drug resistance associated protein 1, and unfortunately it isn't as well investigated as others, like PGP, for example. One reason being that it isn't as highly expressed in the intestinal barrier or the liver, but it is the most highly expressed ABC transporters in lung cells, although usually ubiquitously expressed throughout the whole body, as it is important for the glutathione detoxification process of cells. MRP1 is sitting more or less at the basolateral side of the lung epithelium, meaning that it doesn't face the epithelial side with the air. The air comes into the lungs, but is on the opposite side towards the capillary. And the name gives it away as well. It is often involved in cancer resistance processes, where a cancerous cell would overexpress MRP1 or other transporters to protect itself against anti-cancer agents, which are often substrates to these. Lastly, mutations in the gene of MRP1 are associated with the disease severity of the typical inflammatory lung diseases like COPD and cystic fibrosis, meaning that MRP1 is quite important and has a high function in the lung. Some previous results, we have been collaborating with Oliver Langer from the Medical University of Vienna, and they have been using a compound called MPG and aerosolized this into the airways of alive rats. And this compound was radio-labeled, and they were then able to follow the radioactivity and its distribution through the rat body over time via a technique called positron emission tomography. This is a decently novel technique which allows to visualize the radioactivity non-invasively without taking samples from the bloodstream even. And what they found was that the radioactivity was cleared from the lungs in wild-type rats quite quickly here, the top row, while it was retained in the lungs of MRP1 knockout rats significantly longer. And the AOC and area under the curve values are in line with this, and this study was quite influential because it was the first one visualizing the function of transporters, MRP1 more specifically, in the lungs of an alive organism after application of the compound via the airways. Now, we are interested in the interaction of MRP1 with clinically relevant drugs, and in line with the 3R principle, we are going back into an in vitro model, and we generated a knockout clone of the MRP1 transporter in the NCI-H441 lung cell line. Now, what do we need a knockout model for? Transporters have overlapping substrates and inhibitors, and nothing there is ever truly specific for just one. And only with a gene alteration we can achieve to look at a single transporter isolated after knocking it out while everything else in the cell stays intact. And why did we use our H441 cells and not just an overexpressing cell line and an MRP1 lacking control? That's because our H441 cells are an organotypic cell line. They are derived from lung cells, and even though they are cancerous, they are expressing a variety of transporters, making this model quite physiological, or at least as physiological as an in vitro model can be without increasing its complexity too much. Furthermore, we need these cells to form tight junctions and confluent monolayers to be able to use them for bidirectional transport studies. And lastly, our H441 cells show MRP1 expression levels that are quite similar to primary cells derived from human lungs. So these cells, we took them and generated a knockout clone where the CRISPR-Cas system, the agents were delivered by electroporation, and successfully transfected clones were selected via puromycin. And in the 8 initially generated clones, we saw a complete lack of MRP1 in all of them. And for criteria that are better explained on the poster, the M2 clone was chosen for further evaluation via Sanger sequencing. We looked at the area of interest in the MRP1 gene here and found a perfect alignment of the nucleotide sequence with our study design. This clone was then ready for functional studies, and here we chose a relatively simple release study setup using a compound called carboxyfluorescein. Briefly, how do these work? We don't have our cells on transwells, but at the bottom of 24 well plates in this case, growing submerged in media to which we add the compound carboxyfluorescein, or in this case the prodrug. We load them like this for 1 hour, take off the supernatant and replace it with fresh buffer into which the cells release the carboxyfluorescein, and we take samples from this at a 15-minute interval. And what you get out of this is a cumulative release over time graph here, where we have the wild-type cells in blue and our knockout cells in orange. And what came as a surprise to us at the time was that we couldn't see any difference between the two. Neither in the control group at the top nor at the 2 at the bottom, to which we added a chemical inhibitor as a control for inhibited MRP1. And we hypothesized at the time that this might be due to overexpression of other transporters to compensate for the lack of MRP1. And we investigated MRPs 2 to 9, because they're quite similar, but also organic anion transporters, as they were described to be transporters of the carboxyfluorescein. And while some of these are slightly upregulated and some are slightly downregulated, none of these differences was great enough to explain the complete lack of difference here in this release study. So we switched our substrate to MPG. This is the same one that was used in the in vivo studies I talked about earlier. And why didn't we use this to begin with? MPG, unfortunately, the bioanalysis is just way more complex, and carboxyfluorescein is easy to quantify via fluorescence. And until this point, there was really no reason not to. With the MPG, we were finally able to see a huge difference between the groups, and the compound was only released from the wild-type group, while our KO cells here are at the bottom and approximately at the same level as the wild-type group where we added the chemical inhibitor, meaning that this experiment finally proved our knockout on a functional level, but more importantly, it showed that the carboxyfluorescein apparently isn't a good substrate to study MRP1 in a cell line that expresses a wide variety of transporters, which would certainly be the case in the normal human lung. And this is problematic because we've been using this in the past, but more importantly, many groups are still using this to date to investigate the MRP1 expression levels in cancerous cells in the clinic. Now, the last thing we needed to do for our clone was to assess its ability to form tight junctions and electrical resistances, or rather, if they had retained this ability after gene alteration. And we compared it again to wild-type cells and found no difference, luckily. Both groups were able to form TR values of around 800 ohm times square centimeter, meaning that these cells were ready for bidirectional studies, which is exactly what we're going to do in the future, because we want to assess clinically relevant compounds, but also experimental drugs and their interaction with MRP1. And if we see any hits here, we are going back into an animal model, or rather into an ex vivo model, where we want to use precision-cut lung slices from wild-type and MRP1 rats, knockout rats, to get a better picture at the whole lung tissue and not just the epithelium. Thanks, and if you're interested in the data, there's a lot more on the poster.
Thank you, Johannes. That was very interesting, well, very educational for me in particular. Do we have any questions from the audience, from people who are familiar with these kinds of techniques? While we're waiting, what are the first sort of candidate drugs that you've got in mind to apply to this model?
Good candidates would be something like budesonide. There's indications that this might interact with MRP1. Other than that, ciprofloxacin, for example. So we're mostly looking at stuff that is actually approved for inhalation. There's only like 20 or a good few more maybe. So these would be like the prime candidates. Formoterol example as well.
Still looking around, this must have some interesting preclinical relevance to people in terms of helping us. You mentioned the 3 hours earlier. What way do you think this will reduce our reliance on rodent models and others?
Yeah, I think the big novelty here is the ease of creating a knockout model. For this transporter specifically, but this obviously opens the door to just create knockout models of anything you're interested in. And yeah, the CRISPR-Cas just allows for that. And with the animal model, obviously you see the whole thing in vivo, which is very nice. So you can see it in a live organism. It actually has a function in the lung here. And then with the cell line, we're just able to have a higher throughput of compounds to screen.
If there's no further questions, I'll say thank you very much and good luck.